on Genetic Education podcast Raw and Real, we have a young scientist, Dr. Let us know on which research project currently you are working on.
So currently my area of research is the development of the major cause for the cervical cancer is but we found that all the women diagnosed with the cervical cancer are
So you are actually finding the, we can say, landmarks in our genome, which possibly cause.
so what my study during my PhD I worked on intellectual disability and what my study focused on is it actually triggers massive coordinated upregulation of a gene. I think this is the very important question for the modern geneticist or molecular biologist like us.
Today on Genetic Education podcast Raw and Real, we have a young scientist, Dr. Mansi Desai. Mansi, welcome and thank you so much for your prompt response for the podcast shoot.
Thank you, Tushar, for inviting me.
Dr. Mansi has published more than 20 peer-reviewed research papers, and currently she is working as an assistant professor at the Department of Biological Sciences, CHARUSAT. Okay, so let's begin the discussion, Mansi,
Yes.
and start with something simple. Please let us know on which research project currently you are working on.
Okay. So currently my area of research is the development of susceptibility biomarkers for the cervical cancer.
Okay. So can you tell us more about what you are doing and what is the process and what would be the outcome for the society?
Okay, so cervical cancer is one of the most prevalent cancer in India and it is a second most common cancer diagnosed in a woman. Uh, the major cause for this cervical cancer is a persistent infection of human papilloma virus. But we found that all the women diagnosed with the cervical cancer are not actually HPV infected. And many women who found HPV infected are not developing cervical cancer.
Okay.
So there can be a person's genetic or a person's genetic makeup which can be responsible or which can make person susceptible for the development of cervical cancer. So we generally do is we identify these mutations which are present in a person's genome which can make a person susceptible or a high risk for developing cervical cancer. So our main focus is to identify genomic biomarkers and a transcriptomic biomarkers.
So you are actually finding the, we can say, landmarks in our genome, which possibly cause the cancer infected
Yes.
by the HPV, right? Because not all the HPV infection leads to the cancer.
Yes.
Interesting. So, as I know, you have done extensive research on mental retardation and intellectual disabilities. So, I want to know what are the findings of your research and please shed light on the genetics of mental retardation.
Okay. So when we talk about the intellectual disability, so intellectual disability, commonly known as a mental retardation, are generally divided into two categories: a syndromic intellectual disability and a non-syndromic intellectual disability. So syndromic intellectual disabilities are the categories where a specific features or some distinct features are present along with the intellectual disability. So we can recognize that particular syndrome by seeing or by observing a particular features in that individuals, like Down syndrome. Okay. And other category is a non-syndromic intellectual disability, where only symptoms we observe in a person is intellectual disability. So we cannot recognize a particular syndrome if it is present in a person. So what my study during my PhD, I worked on intellectual disability, and what my study focused on is a specific mutations in a gene, whether it increase a person's risk of developing intellectual disability, a child's risk of developing intellectual disability. So I basically focused on the genes involved in the folate metabolism pathway. So particularly the genes which we studied was MTHFR, that is the methylene tetrahydrofolate reductase, MTHFD1, that is methylene tetrahydrofolate dehydrogenase, and which goes for the protein methionine synthase. So we studied a single nucleotide polymorphism if it is associated with intellectual disability. Along with that, I explored another gene, CBS gene, that is the cystathionine beta synthase gene. Okay, so I studied a particular insertion mutation, and what we found is the MTR, a particular mutation, that is the substitution of adenine at the place of 2756 nucleotide to guanine, was actually more prevalent in children with intellectual disability as compared to the healthy children.
Okay, so that mutation is common among one particular type of category or like many? For example, there is autism, there is Down syndrome, and then
No,
there are many more.
no. That was not a particular syndromic intellectual
Okay,
disability. So that mutation was more prevalent in a children which are of categories of non-syndromic intellectual disability.
okay, okay. So you have done so much research on metagenomics. I guess there might be some projects are also ongoing. So what one thing you find very interesting or fascinating about microbes?
Yes, I have worked on metagenomics. So metagenomics is a field where we study the whole microbial community. The most surprising thing which I observed about the microbes is the plasticity and rapid adaptation, or I will say a rapid functional adaptation of a microbes in response to a particular environmental changes. How quickly the microbial community could change its entire biochemical factory when subjected to a new dietary intervention.
Okay.
We often think that our genes are fixed, but the metagenome is incredibly dynamic. The shift in diet didn't just cause a minor change in the abundance of few of the known bacteria, but it actually triggers massive coordinated upregulation of a gene specific for a particular metabolic path. So it can turn on entire new set of tools which deals with these new food sources.
So
Okay.
these microbes are like, okay, continue.
Okay. So the gut microbiome is not just an a static organ. It's not just a static organ. You can say it is a real-time programmable interface which rapidly translates environmental signals like diet, antibiotics, or stress into some kind of biological outcomes like antibiotic resistance. So maybe this functional plasticity of our gut microbiome help us to widely accept different lifestyles. That means whatever signals that we are providing to our gut, like whatever food we eat, where we are living, what is the environment, based upon that, the microbes are quickly adapt to those conditions. Yes. And can develop in a new form based upon the requirement. And one example for that is the antibiotic resistance. And I guess it's the phenomena that is happening for millions of years in microbes as well. Okay, so let's move to the bioinformatics part. Your work basically is a combination of both wet lab work and dry lab work, right? So it is like balanced approach because most of the researchers majorly focus on the actual lab work, that is the wet lab work. But your work has the balance for both. So what you want to tell to the scientists, how this wet and dry lab work balance is important for research? Like, is it beneficial for like generating more outcomes in terms of research? Okay. So I think this is the very important question for the modern geneticist or molecular biologist like us. So what we generally do is we do a computational predictions. It is also called as a in silico predictions
Hmm.
to filter out enormous amount of data which is generated nowadays. Okay. And what we do is we are doing a particular genomic research. Let's say we are analyzing particular patient's DNA, a particular cancer patient's DNA. So when we analyze its genome, we found thousands of variations. Now, if we will study the consequences of all these thousands variations which are reported in person's genome through a traditional wet lab experiment, it will take years
Hmm.
and millions of money. So what we generally do is using the computational tools, we filter out those signature mutations or those candidate mutations which actually maybe using molecular docking or using a network pharmacology studies, and ultimately we filters nearly 5, 10 to maybe 20 such candidate mutations, and now we study these 20 candidate mutations, or we validate these 20 candidate mutations using traditional wet lab experiment. So ultimately it is saving our time. If I will
Hmm.
give you example in case of drug discovery. So in case of drug discovery, instead of physically synthesizing thousands of different chemical compounds and testing them one by one using some wet lab experiment, if we can short or if we can filter through virtual screening which particular compound can best interact with our protein target, so ultimately it will save our time
Hmm.
and save our money, because wet lab is actually very expensive and time consuming. So to use a computational approach may save your time and your money as well.
Okay. So,
Ultimately, what we generally do after screening the best interacting compound, we can try it using the wet lab experiment. What
hmm.
we found, like in my previous study, we screened some of the genetic polymorphisms may be present in MTHFR or MTHFD1 genes, and using a computational tool, we predicted it to be most deleterious. But when we tested it using our wet lab experiment, we could not find any significant difference in cells functioning. So computational tool can be used to filter your research, but ultimately it should be validated with the wet lab experiment.
So what I understand is it is like you select a topic, then you select a set of genes, then you perform computational analysis where you sort out some common candidate mutations that are there in the candidate genes, and then based upon that data, you start the experimentation in the lab. Okay. Further, at a treatment level, it will also help to like study the effect of various treatments or chemical on a particular target as well. So this is a balance from both the sides. We can say research work, it is also needed during the pre phase of the research and the post phase of the research as well. Interesting. Okay, so the last question is any advice for the future generation scientist or those who want to enter in the science based upon your experiences?
Okay, so one advice I will give to the students or early career researcher like me is to believe yourself, obviously, and to remain active in any situation, because many times situation will be like particular area or interest which you are passionate about to work on, it will not be immediately available to you people. So if you will remain active, if you will keep yourself engaging in a different areas or different opportunities which come in front of you, so every situation will make you to learn something.
Okay, so you have to continuously finding yourself doing novel work. and not to stick on one topic. Okay, okay. Okay, so thank you so much, Mansi. These are the set of questions that we want to ask. We are celebrating science at Genetic Education, and we are discussing science with young minds like you and seasoned scientists as well. So thank you so much for giving us time, sharing your knowledge and research work. It will help students a lot more in their career. Thank you so much once again.
Thank you, Tisha.